Yes, we were discussing about the mutagens. Okay, mutagens, or in other word, we can say the mutagenic agents. What is mean by mutagenic agents? Mutagenic agent means these are the substances that induces the mutations artificially. Okay, these are the substances which induces the mutations artificially, and these are called as the mutagens. Okay, here the mutagens, these are classified into the two groups. The first one is the radiations, and the second one is the chemicals. Okay, radiation and the chemicals. Radiations means what? Radiations means the movement of energy given by the radioactive isotope is referred as the radiations. Okay, movement of energy given by the radioactive isotope is referred as the radiations. Okay, and the treatment of radiation to the plant material, it is called as the irradiation. Okay, and sometimes when the treatment is given by the different types of the radiations, and when the mutations are artificially induced by using the different types of the radiations in the plant, then the new variety is produced by using this method, and that one is called as the this method is called as the irradiation breeding. Okay, it can be defined as the creation of mutations by subjecting the plant to the ionizing radiations and production of the new varieties. Okay, the creation of mutations by subjecting the plant to the ionizing radiations and the production of new variety, it is referred as the irradiation breeding. Okay, it is referred as the irradiation breeding. Okay, here you can see the radiations. These are divided again into the two groups. The first one, the ionizing radiations. The another one is the non-ionizing radiations. Ionizing means that is they produces the ions. Okay, ionizing, here you can see the different types of ionizing radiations. Number one, alpha rays. Number two, beta rays. Then x-ray, gamma rays, and the neutrons. Okay, and the non-ionizing radiations, these are the ultraviolet rays, or we can say these are the UV rays. Okay, and here the second type of the mutagens, that is these are the chemical mutagens. Different types of the chemicals are available which are capable to induce the mutations in the plants, and these are said to be the chemical mutagens. Okay, the first of all, we are going to discuss about the ionizing radiations one by one. Okay, here you can see the first radiation which is used in the artificial mutations is the alpha rays. Alpha rays, that is these are the radioactive rays made up of two proton and the two neutrons. Okay, these are the radioactive rays made up of two protons and two neutrons. Okay, as these are made up of two protons and the two neutrons, that means these alpha rays are positively charged. Okay, these are positively charged and these are emitted by the isotopes of the heavy elements. Okay, these are emitted by the isotopes of the heavy element, but when they passes through the matter, they produces the strong ionization. Okay, when they passes through the matter, produces the strong ionization. But as these are positively charged, when they penetrate through the matter, these are opposed by the negatively charged ions. These are opposed by the negatively charged ions in the matter or in the living tissue, and that's why these are usually having the less penetrating power in the living tissue. Okay, I am repeating this one. As the alpha rays, these are positively charged, and whenever these are passed through the living tissue, then these positively charged ions, these are readily slowed down due to the negatively charged ions in the matter or in the living tissue, and that's why they show the less penetrating power in the living tissue. Okay, then the alpha rays, these are responsible for the chromosomal aberrations. Okay, that means these are responsible for the changes in the chromosome structure. Okay, changes in the chromosome structure. Alpha rays, these are mainly responsible for the chromosomal aberrations. These are changing the structure of the chromosome. Okay, that is about the alpha rays. Then second one here, beta rays. Beta rays, the radioactive rays, these are also the radioactive rays with the negative charge. These are the radioactive rays with the negative charge. Okay, and beta rays, these are nothing but the high speed electrons which are emitted from the nucleus of the unstable atom. Okay, beta rays, these are nothing but the high speed electrons emitted from the nucleus of unstable atom. Okay, but the beta rays, when compared with the alpha rays, these are of less ionizing power. Okay, when compared with the alpha rays, these are less ionizing power, and these are also having the less penetrating power because these are negatively charged, and whenever these are passed through the living tissue, then these are slow down due to the due to the opposition of the positively charged ions in the living tissue. But when we compare the penetrating power of beta rays with the alpha rays, these are having more penetrating power than the alpha rays. Okay, beta rays, these are more penetrating power compared in comparison to the alpha rays. Then the beta rays, they show the variation in the penetration. Okay, they show the variation in the penetration. Then here you can see the phosphorus 32 and the sulfur 35. These are used as a source of the rays. These are used as a source of the beta rays. Okay, that is these chemicals, these are available in the market. Okay, and from which that is the solution can be prepared, and the desired treatment given to the plant. Okay, that is these are available in the market in the form of chemicals, out of which the solution can be prepared and the desired treatment to the plant can be given. Okay, then beta rays here, these are also mainly responsible for the chromosomal aberrations and the gene mutations. Okay, beta rays, they cause chromosomal aberrations and the gene mutations. Okay, then here the another mutagenic agent is the X-ray. Okay, X-ray, these are the electromagnetic radiations. Okay, X-ray, these are the electromagnetic radiations having wavelength much shorter than those from the visible light. Okay, X-rays are magnetic radiations having wavelength much shorter than those from the visible light. Okay, here we can see the source of the X-ray. Source of the X-ray is the X-ray machine which is installed in the room. Okay, that means if the plant breeder wish to give the treatment of X-ray to the plants, then the plant breeder have to take these plant to the room where the X-ray machines are installed. Okay, that's why this is also the limitations of using this method, because if there are large number of the plants and the plant breeder wish to give the treatment, then he have to take all these plant to the room where the X-ray machine is installed. Okay, the source of X-ray is the X-ray machine which is installed in the rooms. Okay, the average wavelength of the X-ray is the zero point 0.5 angstrom. Okay, these are having the less wavelength than that of the visible light, and the mainly the effects of the X-ray, these are due to the sparsely ionizing electrons. And also the X-ray, these causes the chromosomal and the gene mutation. Okay, these are responsible for the chromosome and the gene mutations. Then the another one, the gamma rays. Gamma rays, these are also the electromagnetic radiations. These are similar to the X-ray in their physical characteristics and actions on the organisms. Okay, gamma rays, these are the electromagnetic radiations similar to the X-rays in their physical characteristics and actions on the organisms. And that's why the gamma rays, these are also referred as the natural X-ray. Okay, the gamma rays, these are the natural X-rays of the short wavelength. Okay, these are having the much shorter wavelength when compared with the X-ray. Okay, and the wavelength of the gamma ray, it is 0.01 angstrom. Okay, which is very less compared to the X-ray and the visible light. Okay, and as the gamma rays, these are having very short wavelength, that means these rays are having more energy, and as these are having more energy, that means these are having more penetrating power than X-ray and the visible light. Okay, as these are of short wavelength, that means these are having the more energy, and that's why these are more penetrating than X-ray. Okay, here the gamma rays, these are produced by the radioactive decay of the certain chemicals like the radium C14 and the c carbon 14 and the cobalt 60. Okay, these are produced by the radioactive decay of the certain elements, for example radium C14, that is the carbon 14 and the cobalt 60. Okay. The treatment of the gamma rays, it is given in the specialized designed gamma garden. Okay, the treatment of gamma rays, it is given to the plant in a specialized in specially designed gamma garden, and they mostly produce the gene mutation. Okay, they mostly produce the gene mutations. Okay, then the next one, the neutrons. Okay, neutrons, as the name suggests, these are the electrically neutral particles. Okay, these are electrically neutral particles, and the biological effects, these are mainly due to the densely ionizing protons, which may be fast as well as slow. Then the neutrons, as these are electrically, where they are penetrating, these are not opposed by the positive or the negative charged ions in the living tissue. That's why neutrons are highly penetrating. Okay, these are highly penetrating, and the source of the neutron is the nuclear reactors and the generators. Okay, generators and the nuclear reactors, which are available only with the atomic energy department of government in the different parts of the country. Okay, the source of the neutron is the nuclear reactors and the generators available with the atomic energy department of government in different parts of the country. Okay, then the non-ionizing radiation, here the UV rays. Okay, up till now we have discussed the ionizing radiations, but this is the non-ionizing radiation, ultraviolet radiations. Okay, these are the electromagnetic radiations of the wavelength shorter than the visible light. Okay, these are having the wavelength in between 100 to 300 nanometer. Okay, 100 to 390 nanometer. UV light, these are shorter than that of the visible light. Okay, the source of the UV light, these are produced by the mercury vapor lamps or the tubes. Okay, mercury vapor lamps or the tubes are available, which are the source of the ultraviolet radiations. Okay, then these are the low energy radiations and not cause the ionization. As these are low energy radiations, the ions are not produced, and these are having very less limited penetrating capacity. Okay, these having very less limited penetrating capacity, and that's why these are not efficient in producing the chromosomal and the gene mutations as compared to the ionizing mutations. Okay, whenever we compare this non-ionizing mutagenic agent ultraviolet radiations with the ionizing radiations like the alpha rays, beta rays, gamma rays, X rays, then these are not efficient in producing the chromosomal and the gene mutations. Okay, but the ultraviolet radiations, these are used to study only the pollen grains. Okay, as these are not efficient in producing the chromosomal and gene mutations as compared to the ionizing mutations. The ultraviolet radiation, these are used mainly to study the pollen grains only. Okay, that is all about the ionizing and the non-ionizing radiation. Then the different types of the chemical mutagens, here you can see, which are used to induce the mutation artificially. Okay, the first one is the alkylating agents. Alkylating agents, the different types of alkylating agents like the sulfur mustard, nitrogen mustard, ethylene oxide, imines, and the diazoalkanes, which are used to induce the mutation artificially. Okay, then the different types of the acridine dyes like the acriflavine, proflavine, ethidium bromide, these are used as a mutagenic agent. Then base analogs like the 5-bromosyl, 5-chlorouracil, these are used as a chemical mutagen. And apart from these three types, there are other type of the chemicals which are used as the mutagens, and these are the nitrous acid, hydroxylamine, sodium azide, colchicine, ethyl methyl sulfonate, and the malic hydrazide. Okay, and the malic hydrazide. Okay, we have discussed the different mutagens which are used in the mutation breeding technique, like the alpha rays. Okay, for example, alpha rays, these are the ionizing radiations, but these are mainly responsible for the chromosomal aberrations. But in the mutation breeding, plant breeder is mainly concerned with the individual gene mutations. That's why generally the alpha rays, these are not commercially used in the mutation breeding. Okay, then second one also we have discussed the beta rays. Okay, these are also having very less penetrating power property, and that's why also the beta rays, these are not generally used commercially in the mutation breeding. Okay, then we have discussed the x-ray and the gamma rays. Okay, these are mainly responsible for the individual gene mutation which are of considerable importance in the mutation breeding, and that's why these are the two methods which are extensively used by the plant breeder. Okay, there is also another method which is we have discussed, the use of neutrons. Okay, but the source of the neutron, it is the neutron, these are also equally responsible for the individual gene mutations, but the treatment of the neutrons is generally given. Treatment of neutrons, suppose that the plant breeder wish to give the treatment of neutrons to the plant material, then he has to contact the atomic department of the government, because the source of the neutron, it is the nuclear reactors and the generations, and these are only available with the atomic energy department of governments. Okay, and that's why it is very difficult to use the neutron induce the mutations in the plant, and that's why the two most popular methods which are used for the inducing mutations artificially in the plants, and these are the x-ray irradiator and the gamma garden. Okay, in the first photograph you can see the x-ray irradiator which is used to give the treatment to the plants. Okay, the only one difficulty in this treatment is that the x-ray irradiators, these are installed in a specialized room. Okay, and plant breeder has to take the plant breeder, whether it is seedlings or the seeds, to that specialized room to give the treatment. Okay, and that is the limitation to use the x-ray irradiator method. But in the gamma garden you can see that is these gamma gardens are specialized garden in the open field. Okay, here you can see in the center a tower is situated, and on this tower generally the source of the gamma rays is kept. Okay, for example, suppose that the cobalt 60, that radio isotope, it is placed in that tower, and then accordingly the plants, that is these are cultivated surrounding that tower. Okay, and then this garden, it is well protected by the wall of bricks. Okay, to protect the other animals or the peoples from the radiations. Okay, and that is specially designed gamma gardens. Okay, that is all about the different types of the mutagens which are used in the mutation breeding technique.